Let me just start by introducing myself. I'm David Spiegel. I'm a professor of chemistry and pharmacology at Yale. I'm also a biotech entrepreneur and been involved with several biotech companies. And at the beginning of this sort of COVID crisis, started to become interested in testing, in part because of my good friend Eric Gordon, who's now my business partner, was one of the early COVID patients, and so the testing for him was an issue very early on, and so we made a number of connections and got involved with with several different antibody kits, and found one that we're going to talk about today that we believe is the best, based on some studies that we've done at Yale and elsewhere. So this is about a multi-site validation of this SARS-CoV-2 IgG IgM antibody detection kit. We published the paper on, or we posted the paper on medRxiv, which is a sort of publicly available server for pre-printed, non-peer-reviewed manuscripts. And just want to thank all the people who were involved in this. It kind of took an army to both collect the data and analyze the data, but we're very pleased with the results. So I think SARS-CoV-2 and COVID-19 needs no introduction. Obviously, it's an incredibly serious, severe disease. It's affecting a large number of people worldwide. We have millions of cases globally, hundreds of thousands of deaths globally, and there needs to be lots of diagnostic testing to keep the disease under control. And also, of course, we're all hoping and praying for new kinds of treatments. The lateral flow assay, the test that we're working with, is a lateral flow assay. But before I talk about the test, I just want to mention how useful antibody tests are going to be. They're really useful for several reasons. Lots of people ask me, why an antibody test? Why not other kinds of tests? I think there are three reasons. One of them is the test tells you your immunological status. That is, are you protected by your immune system? And this will aid in decisions about whether or not to go back to work, for example, or whether, for employers, whether to bring people back to work. It's also important secondarily, or to monitor public health statistics. So understanding who has been exposed to the virus and who has immunity, of course, on a population-wide scale is extremely important. And then thirdly, it's sort of funny, you know, I haven't talked to anybody who hasn't at some point during this last several months thought at some level that they may have had the disease. Everybody's curious about whether or not they were exposed. And so this test will satisfy that curiosity. And I think everybody is really going to want one of these tests. And so we're very excited to bring them now to people, to patients. The tests are incredibly easy to read. So the lateral flow assays, which means they're like pregnancy tests, they have a very simple visual readout. They don't require any special optics or anything, instrumentation. They give you a band if they're positive. And then there's, in this particular test, there are 3 different bands that are possible. One that's called M for an antibody type called IgM, another one called G for an antibody type called IgG, and then there's a control C that tells you if the test is working. So if you have a band for M, means you have IgM antibodies. For G, you have IgG antibodies, and that's it. Test interpretation is extremely straightforward. It also happens quickly, so the test is incredibly easy to interpret, and it gives results in actually less than 30 minutes from start to finish, but you'll see later you can see results in as soon as 30 seconds. Let's talk about the clinical trial that we did. We have a pretty sizable number of patients. We looked at a total of 5 different locations: Yale New Haven Hospital, which is the study that I oversaw, and then the manufacturer, our collaborator, BioHit, in Hefei in China looked at 4 different sites. And these included hospital sites as well as these Chinese CDC sites to give a total number of 1,892 patients in the study, approximately equal number of positives and negatives. So the Yale study is what I'm going to dive into most deeply. We've collected a whole lot of sort of data analysis on this study. What we see here is an age range that's very well representative of the adult population, ranges from 26 to 92 years. Genders are essentially equally represented, and the infection range, there's this, or infection time, there's this variable that we've come up with, which is defined as the number of days from patient-reported first symptom to the date that the sample of blood that we're looking at in the test is collected. So if you have a symptom on Monday and you collect the blood on, say, Friday, you have the infection time of 4 days. And so this is the variable we're looking at here. And again, we have a wide range of infection times. And infection times are very important, as you'll see, because the immunological response is proportional to how long after you were exposed to the virus it is before we take the sample. So just to cut to the chase, I want to talk about how the test performs. So the test performs extremely well across all the sites we looked at. We combined all the Chinese sites together and have the Yale sites separated out. What we see here is that, and here we're looking at positive samples, we see for patients greater than 14 days, essentially close to perfect sensitivity, which means we're picking up true positives and eliminating false negatives at a rate of essentially somewhere between 94 and 100%. As you'll see in this range between 1 to 14 days, the sensitivity of the test goes way down, and that's because patients haven't produced the antibodies that the test is measuring at as high of a frequency in that range. You'll also notice that there is some difference, substantial difference between the 1 to 14 day sensitivity for the site in China versus the 1 to 14 day sensitivity for Yale. And that's because we conducted the study slightly differently. So at Yale, we pre-selected the patients to have antibody already for a measure of how well the test is detecting antibody. The Chinese sites didn't do that, which is actually consistent with what the FDA is recommending nowadays. And so this difference between 45.6 and 77, for example, is the result of lots of the patients in the Chinese study simply not having antibodies at all, whereas all of our population had antibodies. And that was detected using a gold standard technique called ELISA. ELISA is often used in laboratories. It's sort of a complicated laboratory test to determine if a patient has antibodies. And ELISA in this study served as the gold standard. Let's talk now about the negative samples. Probably the most important parameter of this test is its ability to distinguish true negatives from false positives. So in other words, we don't want people thinking that they're immune when they're actually not. And specificity is sort of the percent of patients that are true negatives. And so what we're seeing here, in other words, when the test result reads out as a negative, it's the percentage of the time that the patient actually is a negative. And so what we see here is for both IgG and IgM, the specificity is close to 100%. Which means the test is performing almost perfectly across a wide range of samples at a wide range of different sites. We also developed something called the visual inspection score, where what we can do is visually inspect the tests, and based on the darkness of the bands, you can see 42 shows no bands for G or M, 43 shows very faint bands, 44 shows sort of intermediate strength bands, and 45 very dark bands. we stratify the results based on sort of weak, intermediate, and strong positives, and that gives us what we call a visual inspection score, which we'll talk about in a moment. But you can also see from this picture the tests are incredibly easy to read. It's very clear when the test is reading out a negative versus when the test is reading out a positive in most cases. So here we use this infection score to determine two things. First thing, if you look at these graphs, what we're showing here is the amount of antibody now measured by this laboratory test called ELISA on the y-axis as a function of infection time, how many days from first reported symptom to when the test is taken. And what we see here, if you just look at these graphs and pretend there's no color in the dots, what we see is essentially that the number of days, the infection time is, the antibody response is proportional to the infection time. And this is known, we're not the ones who discovered this. What it's saying is that the longer out a patient, farther out a patient is from exposure to the virus, the higher the chance it is that their immune response will generate antibodies to that virus. And it's antibodies that our test is measuring. Now, if we incorporate these colors, what we see is red is our strong positives, orange is our intermediate positives, and the yellow is the weak positives in the visual infection score I just showed you. And what you see here is that couple things. First of all, there are strong positives. The reds are found throughout infection times, but maybe most interestingly, the weak positives, intermediate positives, as well as the false negatives, which are the gray circles, are all clustered towards shorter infection times. And that's telling us that, again, the test is most likely, or in this case, the test is most likely to read out as a negative when patients have very low antibody levels, which correlates with short infection times. And that, of course, makes sense. Lower the antibody levels, the less likely the test is to pick them up. We also looked at this visual intensity score with respect to antibody titer. So in other words, if we look at the different the scores that we're assigning, are those scores, are the stronger positives essentially correlating to patients who have stronger ELISA responses? And the answer is yes. You can see that there's a sort of distribution that increases, that the threes tend to have more antibodies, both in the IgG and the IgM channel. We can also look now at the differences in antibody levels in the bottom panel for the false negatives versus the true positives. And again, what you can see here is that the true positives tend to have higher antibody levels in both the IgG and the IgM panel. Again, suggesting that the test is quite sensitive and that it is reading out essentially proportionally to antibody levels. So this is actually an interesting finding because people often think of these lateral flow assays as sort of binary readouts, either you're positive or negative. But what this is telling us is that in addition to doing a good job of distinguishing the true positives from the false negatives, the visual intensity score actually seems to be measuring antibody concentrations. The more intense the line is, the higher the antibody concentration in your blood is likely to be. We then went ahead and looked at what we call an automated intensity score. And what we did here was we took pictures of the test with a smartphone and then used the computer algorithm to measure the intensity of the bands computationally. What we see here is actually pretty interesting. We see on the top two panels, you can see for both IgG and IgM, that again, this automated intensity score is doing a pretty good job of reading out antibody levels. On the y-axis, we have the ELISA antibody level. So essentially, our automated intensity score is proportional to the antibody level that the laboratory test is reading out. And then furthermore, we see really excellent agreement between the visual intensity score and the automated intensity score, noting that, again, they both probably are good ways to measure the results of these tests. But what we see, and this is not shown here, is that there is some overlap between the visual intensity score, oh, well, you can see this here, between the visual intensity score and the automated intensity score for the lower intensity bands. And what we take from this actually is that the visual intensity score may actually be more reliable. In other words, a person's ability to distinguish the very faint positive from the absence of a band, the true negative or the false negative, depending, any negative, actually seems to be more reliable than the computer's ability to do that, at least in this study. And so we're of course looking to improve the automated intensity scores so that the computer can do a better job in terms of the cutoff for positives versus negatives. But for now, based on our data, we're suggesting that these tests be interpreted by an expert, someone who knows how to read them, even though in fact anybody can read them. For the stronger intensity bands, the eye does seem to do a better job for the weaker intensity bands. We did a number of other studies just showing how versatile these tests are and how robust they are. We looked at different kinds of blood products. We looked at whole blood just from finger stick as well, or actually this is from venous blood and serum, and we see 100% of the assays perform identically between whole blood and serum. There's no change between those two. We also looked at the time course of the band development on these lateral flow assays, which is actually quite interesting. Again, you'll see, even though the tests we recommend the test to be read out at around 10 minutes, what you see is that in this case, you can see the band starting to form as early as 30 seconds. You see pretty clearly that these bands are in place for the G and the M after 2 and 5 minutes. And after about 10 to 15 minutes, the intensity of the band in the background remains stable. Actually, we looked at 2 hours, 12 hours, 24 hours. The bands are quite stable. But we do recommend just reading between 10 to 15 minutes and not paying attention to what happens before that or afterwards. We also looked at varying the amount of the blood that we add to the test. And so it's obviously possible for someone to take the test and add much less than the recommended 10 microliters, or in some cases much more. We looked at a wide range down to several orders of magnitude, both above and below the recommended value here, which is 10 microliters. And what we see is that the band intensity is actually quite stable over between 2 and 4 orders of magnitude. That's essentially over between 100 and 10,000-fold errors in the amount of blood. So we were actually quite surprised by this, that we can see positive test results really across very small amounts of blood, and the same positive test results, very large amounts of blood. We looked at 2 different patients here, patient 10 and patient 1. Patient 1 is sort of a weaker intensity band. Patient 10 is a stronger intensity band. And again, we see this result maintained over a wide spectrum of blood amounts in both. The other thing that we did here is to look to check and make sure that both the G and the M bands are actually reading that particular type of antibody. I'm not going to go into detail here. Not sure if there's but in terms of the science of it, we're able to deplete selectively the IgG antibodies from whole blood. And what we see here is that we start out with a very intense sort of series of bands on the left-hand side. And what we see on the right-hand side is when we selectively deplete the IgG, we're seeing only now a band for IgM. And so, again, this is telling us that the test, the M band, is actually detecting IgM, and therefore also the G band is detecting IgG. So the tests are sort of true to their claim. So let's just conclude, and I'm very happy to answer any questions. I realize not everyone in this audience is a scientist, But just to overview, we found that our lateral flow assay performs with excellent sensitivity and specificity across multiple sites, actually both at Yale and in China. And we've looked at now a wide range of patient characteristics, and the test is quite robust. The sensitivity, as I mentioned before, the ability to detect true positives varies with infection time and also with antibody level, as I said, versus ELISA as a gold standard. What we are measuring here is patients with shorter infection times have lower antibody levels, and therefore the sensitivity goes down. But greater than 14-day infection time, as we discussed, we have a 94+% sensitivity. The visual intensity score, which we developed, does seem to perform better than the automated intensity score, although they're both performing quite well, suggesting a role perhaps for telemedicine in reading these tests. The bands develop very quickly, as you saw, and are sensitive to a wide range of serum dilutions. The bands in the tests are specific for IgG and IgM, and we believe that the tests are appropriate for point of care administration in a doctor's office or a CVS or a pharmacy. We've also been in close conversations with the FDA, and we got some very good news this week that our results are viewed favorably by the FDA, and that we are very close to receiving an EUA. We expect to be able to sell these tests within the next week. So very grateful to all the co-authors, very grateful to all of you for your time and attention. I'm very happy to answer any questions. Thank you.